With today's busy lifestyle, it's easy to become stressed and tired, but the right multivitamin can help you stay performing at your best. Try the DS range. Designed to help support your active lifestyle, DS Sport has the right combination of vitamins and minerals to give you the extra energy you need to perform at your best. Plus, it's available in sachets for on-the-go convenience. For the energy you need to tackle your busy lifestyle, try the new look DS range. This has been Medifax for DS24.
Good evening, everybody. Very warm welcome to you from a chilly Johannesburg winter evening. On behalf of Wits Sport and Health, as you know, this is a clinical and research network that is coordinated by the Faculty of Health Sciences at Wits University and really designed to pull together all the expertise in sport and exercise medicine around the greater Johannesburg area. And tonight's webinar is hosted specifically by the Concussion Interest Group. We have a number of interest groups and we encourage each group to host 3 or 4 webinars a year. And we're very pleased to host our 3rd concussion webinar for the year. A special welcome to all our regular attendees, but also to some guests from overseas. I know we have some of our research colleagues from the UK, some of our Irish rugby colleagues have joined, and we've got people from as far afield as Qatar as well. So lovely to have an international field joining us in South Africa today. And of course, none of this can happen without technical expertise. So we thank very much Robin Saggers, Dr. Robin Saggers, who puts together these webinars and promotes them for us, and to the management committee of WiSH as well, together with the social media support that we receive from Nandi Buthelezi at Wits Sport. We're very grateful for that. And of course, to host these webinars, as you saw, we have a very committed sponsor, the Asina Lita Pharmaceutical Group, and we thank them very much for their ongoing support of this webinar series and future academic endeavors as well. We're very grateful to have them on board. So as I say today, it's sports concussion again, and we really are very, very privileged to have a speaker with specific expertise in this field. A couple of months ago, I received a call from Dr. Christos Profyris, and he said to me, "John, have you ever thought about brain mapping and its impact on sports-related concussion?" And although I'd come across it in the literature, I really wasn't that au fait with the concept. And he very kindly sent me some of his presentations and some of the literature. And I found it a very fascinating journey since. And I think you're going to find the same over the next 60 to 90 minutes. As Chris introduces us to some of the concepts behind the Connectome project, behind brain mapping, and perhaps where that might fit into the realm of sports concussion. Because as you know, it's a very difficult diagnosis to make, difficult condition to follow, monitor, and to manage. And this may be an area that actually combines the insight into some of the structural and functional changes in the concussed brain. And that's what excites me about brain mapping. Dr. Profyris is trained, he's internationally trained, let me sum it up that way. He graduated from the universities of Cambridge and Oxford, where he received his medical training. He also received his postgraduate neurosurgical training in South Africa on the Wits circuit, and is a fellow of the College of Medicine in South Africa. And very, very significantly for him is a fellowship that he completed in Australia, where he gained specific insight into brain mapping. And that's what he's going to share with us this evening. So I'm going to leave it there. I'm going to ask you to please think about questions that you can post in the Q&A panel, which we can then post to Chris afterwards. We look forward to your participation. I'm going to turn my camera and sound off. I'm going to hand over to you, Chris, and we look forward to the journey you're going to take us on in the next hour or so. Thanks very much.
Thank you, John. Thank you for the invitation, and good evening, everybody. So today what we're going to talk about is concussion, but in the context of the connectome. So the first part of my talk will be more based on terms of what the connectome is. And then the second part of the talk will go into the specifics relating the connectome to concussion. So if we think of the brain, it is a miracle of creation. And it's a complex structure which defines every single activity we do at every moment of our lives. It's responsible for who we are as human beings, the way we talk, the way we think. It guides our interests, our passions. And it's been a real journey in terms of understanding how the actual brain functions. So when we go back into ancient times where people were starting to understand that it's a seat of thought, there were many theories about how the brain worked. And it went from being a radiator of the heart to the ventricles considered as a seat of thought. But it wasn't until the 1790s where people started thinking about it in the context of an electrical organ. And the reason I'm going into history, because a lot about how we think of the brain today is guided by our history. So in the 1800s, a School of Localizationism came about where we had the famous Phineas Gage. We had an iron rod that went through his orbit into his frontal lobes. And from that injury, people started deducing the function of the frontal lobe because he became impulsive, making bad decisions, becoming violent. So people deduced what the frontal lobe does from accidents such as Phineas Gage's. And the same came about for localization of language, such as the Broca area being the motor part, and Wernicke deducing that a comprehension part of language localizes to the superior temporal gyrus. And that's important because this train of thought has influenced how we understand the brain today, because there was an overwhelming pressure to localize every single function to a part of the brain. And ultimately, this culminated in the most detailed map that we had in medicine and what we've all probably studied in med school when we were studying the brain, known as the Broadman Map, where little numbers became symbolic of what the brain does. We had 47 different areas on the Broadman Map, and he built this in 1909 based on histopathological data and looking at the differences of the cells. And it's a decent map when one considers that it was created in 1909, because we know what the primary motor cortex is, a sensory cortex, language areas, visual areas. But the question really comes, based on today's technology, is this the ultimate expression of what we know as neuroscientists about how a brain actually works? And the answer is no. In the last few decades, neuroscience has, leaped bounds. And while we study a Broadman map or cortices and gyri and think we have a really detailed view of what the brain looks like, the reality is that our current clinical understanding is quite hazy. And this is actually what we're looking at, even though we think we're looking at at a very crisp picture of what the brain is and what a brain map is. And in order to get a more detailed understanding, one needs to consider the concept of the human connectome. The human connectome endeavors to essentially study all the brain's connections and deduce its function. It's a very complex undertaking, purely by the size of the number of cells and components that make the brain. So we have 86 billion brain cells, more than 1 trillion supporting glial cells. And on average, each one of these nerve cells makes more than 20,000 synapses. And then we're passing about 17 trillion electrical signals a second. So when you consider the complexity of an organ such as the brain, it becomes quite overwhelming to study it. But there's different scales of studying. So we might decide to look at the brain on a microscopic level, or we might consider to look at it at a macroscopic level. So what this little graph shows essentially is different technologies that we can use to map the brain, such as EEGs, MRIs, calcium imaging, electron microscopy. The ultimate idea, the ultimate study would be to actually understand how every one of those 86 billion brain cells is connected and what they do. So people have endeavored to do this through electron microscopy. But what becomes very clear when you're trying to study the brain in that amount of details, it's an overwhelming task that won't be completed in our lifetimes. So the C. elegans has 302 brain cells with 5,000 synapses, and White and Brenner endeavored to study the entire connectome of the C. elegans to try and get some insights in terms of the human connectome. What they realized is if you're going to do that and serially slice the entire nervous system and study it under an electron microscope, that it takes about 10 years. So on average, it's estimated that if you were to study 1 millimeter cubed of cerebral cortex, it takes about 800 man hours to process that information on the electron microscope. So if we extrapolate that work to higher order organisms, it would take about a decade to map a mouse brain and a whole millennia to map the human brain. On top of that, complexity arises in this approach because the sheer data that would be produced through these methods would be too overwhelming even for the most sophisticated current machine learning protocols. So it's estimated that the data storage for a human cortex would be 1 zettabyte, which is roughly about the global mobile phone traffic for the entire planet over a year. So clearly that's not practicable. So even though it might produce some interesting science, we can't use this information to influence our clinical practice because ultimately you want to understand the connectome and study it, but it mustn't be this obscure thing that you just hear about in a talk and have no practical applications for it. So what becomes very interesting is trying to study the connectome through functional magnetic imaging or fMRI. Most hospitals have an MRI machine, and most MRI machines in the last decade have capabilities of doing fMRI. So if we build some concepts in terms of studying the connectome with our current technology that exists in our hospital and provide a platform to do that, potentially we can get a lot of very useful information when faced with patients with neurological problems. The heart of this is the data that's been generated from the Human Connectome project. So neuroscience had a lot of interesting individual publications in relation to the connectome. And in 2009, the NIH provided a $40 million grant to the leading neuroscience, neuroimaging institutes of the world, such as University of Minnesota, Wisconsin, and Oxford University. And what they did is they used that funding to scan approximately 1,200 people, predominantly with 3 Tesla MRIs, and also did extensive behavioral testing on those patients. In fact, when they started this project in 2009/10, it took them 2 years before they even scanned the first patient because they had to significantly upgrade existing MRI sequences and hardware. Now, I am going to talk a little bit of about MRI and sequences. So we can look at the brain in different ways by using different sequences. And some of the sequences that are used to map a brain are a structural MRI sequence, a diffusion sequence, and a resting state functional MRI. And I'm just going to talk a little bit more about each one of these sequences. So a structural MRI is simple. It's just a T1 image predominantly, which just looks at the brain structure, the gyri, the sulci, and what it actually looks like. And the reason we need this is to have a backbone to what we pin the subsequent sequences to. The second sequence that's used in brain mapping is diffusion imaging. So what we're doing with diffusion imaging is we're taking advantage of water molecule movement within axons. So our brain is composed by a huge volume of axons, and axons transmit electrical signals. And in doing that, they've got a lipid membrane around each axon where water molecules can move happily up and down an axon. And that's called anisotropic movement, which we'll discuss in a little bit. but they can't cross axonal membranes because the lipid molecules stop them. And I'll show you that in a little bit more detail. The last sequence we use for brain mapping is functional MRI, and specifically resting state functional MRI. So in the next two slides, I'm just going to describe the diffusion imaging in a bit more detail and the bold or resting state functional MRI sequence. So if we look at diffusion MRI, as I alluded to before, we've got axons, which are made of lipid membranes. So water molecules can travel up and down happily within an axon, but they're not able to cross across membranes because they are hydrophobic. So what happens is the specific sequences we use are able to calculate the movement of axons, water molecules up and down an axon, and that's called anisotropic movement. Because by nature, what water molecules do is they move haphazardly, and that's called isotropic. So from doing that, what we're able to do is then start coloring in or finding where these axon tracts are. So in this diffusion image, which I've used for a brain tumor patient, I'm able to visualize the corticospinal tract through this property, a major brain highway called the superior longitudinal fasciculus. And that's all using diffusion MRI. The next sequence we'll talk about, which we use for brain mapping, and this is more for functional brain mapping, is resting state functional MRI. So in general, what functional MRIs do is they use bold signaling, which is a way of tracking blood movement within the brain. So blood has hemoglobin, and hemoglobin has ferromagnetic properties. So when we use parts of the brain, more blood flows to that blood area. And what happens is the ratio of deoxygenated and oxygenated hemoglobin changes, and we're able to use that property to figure out which parts of the brain are active with blood. Now, what most people know is task-based fMRI, and that's an older type of MRI, which has huge applications. And that involves asking a patient in the MRI machine to lift a hand or to talk, and then we figure out which parts of the brain are active based on the task. Now, in '92, there was a PhD student called Biswald, and what he noticed while he was doing these task-based fMRI experiments, that a lot of patients that were just sitting there at rest had very predictable parts of the brain that were lighting up, even though they were doing nothing. So they were just sitting there with their eyes closed, just waiting to be part of an experiment. And this was not random. And that gave rise to resting state functional MRI. So what resting state functional MRI tries to do is identify which networks or which parts of the brain fire together at the same time, and i.e. are connected together to make a single functional network. And what happens is the brain at rest circulates through many different networks, almost like a computer which is resting with its screen off. It's still running, but if you press a button, it's ready to fire. That's the same way we're currently thinking of the brain. We've got different networks that are firing together, and they cyclically rotate on a continuous basis. So what has the Human Connectome project done? Well, in essence, it mapped these 1,200 patients and created an open source database where neuroscientists could extract that data and perform numerous different types of experiments and studies to give us a deeper understanding of how the brain works. One of the big problems that I alluded to in the earlier part of my talk was that the most sophisticated brain map that we are using as clinicians is called the Broadman Atlas, which has 47 areas and was constructed in the early 1900s. So one of the most prominent publications that have come out from the Human Connectome project is a whole new brain atlas. So this publication in 2016 was published in Nature by Glasser, and the father of Connectomics, who essentially is Van Essen. And what they did is they extracted all the Connectome data, and then they built an AI model or a machine learning model, which classified the brain based on cortical thickness, myelination, functional resting state functional MRI studies, as well as previous known parcellation maps. And what they came out with was a hemisphere which has 180 areas. So a brain from 47 areas has gone to 180 areas. And each one of these little areas is called the parcellation. What their study showed was that there was 97 new brain areas that none of us even knew existed before the machine learning told us it's probably a good idea to subclassify it that way. Of the original 47 GLASA areas, only 23 remained in the parcellation scheme. And of those, only approximately 10 remained in their original form. What this did, though, is it's created a whole new map for us to try and describe the brain, and it makes it a lot easier to compare individual brains. So we've got a language now that if we're looking at area 8C or PFM, we can use machine learning to map that area on individual brains. So when we're now starting to compare different brain areas through functional MRI studies, we've got something that's directly comparable between many different patients. So that took the field forward. What else is, the other thing that's filtering through the literature is machine learning based diagnostics, where we can scan patients' brains through that diffusion imaging that I talked about, through that resting state functional MRI imaging that I talked about, and use machine learning to actually start diagnose problems. And I'll briefly talk about depression, because studying the connectome has radically changed mental health and the field of psychiatry. So in this paper by Drisdale in 2016 as well, what they did is they had lots of patients with major depression and lots of patients who were normal. And they had connectome data based on diffusion imaging and resting state functional MRI data. And what they did is they put all this data in a machine learning model, and they asked a simple question: if we use machine learning and this type of imaging, can we tell the difference between patients with major depression and patients who are normal? And the answer was yes, we can. In fact, you can use machine learning to get a sensitivity of almost 85% and specificity of almost 85% in terms of differentiating patients with major depression and patients that don't have depression. And what the machine learning told us further is that depression can actually be broken down into 4 separate diseases that we never even knew about. So the take-home message here is we can combine functional MRI data, integrate it with machine learning, and the machine learning can tell us a lot about diagnostics. So before we go into some of the studies in relation to concussion, I just want to introduce you to the concepts of cerebral networks or brain networks. So the major thing that the Human Connectome project taught us, especially through resting state functional MRI, is that the brain has areas which are wired together, they fire synchronously, and they bring about a common function. There's probably more than 150 brain networks currently known, but if we look at the major networks, which are indivisible, we're probably looking at approximately 7 main networks, and I'm just going to go through them just as an introduction. So we've got 2 main input and output networks, and those are familiar to everyone. So the sensory motor network and the visual network. We've got an ancient network, which if you follow the field of architectonics makes sense as well. And that's called the limbic network. And then we've got 4 main cognitive networks, which unless you've been recent in the field, it might be brand new terminology. So we've got the default mode network, the central executive network, the salience network, and the dorsal attention network, which I will explain shortly. And the reason I'm going through this, something like the default mode network is thought to be very important in concussion and traumatic brain injury. So it's just going to be a brief introduction. Okay, I'll start with what's familiar. So the sensory motor network, when we're looking at resting state functional MRIs, what we notice is that the motor cortex and the sensory cortex fire together as one network bilaterally on both sides. They're interconnected through the corpus callosum and they fire as a single network. And this network also involves the primary auditory cortex. So motor, sensation, and auditory modalities all fire as one. And when you look at the amount of parcellation, so if you're going to study this on a GLASA atlas, and look at how many parcellations are involved, it involves about 30 parcellations. And, you know, this network makes sense. If you're going to move your arm, you probably need to know where it is in space. And that's why everything's firing as one network. The largest network by far is the visual network with 52 areas or parcellations based on the Glasser scheme. And it makes sense. It's all the visual cortical and associated visual cortical areas firing as one. And it's the largest network in humans because we use vision as our primary sensory modality. The limbic network involves the medial frontal lobes, medial temporal lobes, and has 24 areas or parcellations. And that's important for regulating emotion, motivation, fear. And it's a primal ancient network. And, you know, we have evolved from creatures that are just seeking food and avoiding danger. So it's no surprise that this network exists in our brains, and it's situated in the depths of the brains, and it's responsible for these characteristics. Now, the interesting networks, which have many parallels to concussion and are very important in us understanding traumatic brain injury, are our cognitive networks, because ultimately that's what makes us humans, our ability to understand information and process it. So how does our brain actually do that? Well, what became interesting from studying resting state functional MRI was that one of the major networks, and it's also called the task negative network, is the default mode network. And that involves areas in the interparietal sulci, the cingulate gyrus behind the corpus callosum, and the medial frontal lobes, as well as some people look at an extended default mode network and include some areas on the temporal lobe. This has 27 areas on both sides of the brain. And this one's important for internal thinking, imagination, processing our relevance as human beings to our environment. So this is the network when we're in deep thought, contemplating our future, and it's these areas that actually fire. Its exact opposite, which never fires at the same time as the default mode network, is the central executive network. And that involves a lot of the dorsolateral prefrontal cortex, some of the interparietal areas, and some of the posterior frontal areas. And this has 24 areas, and it's responsible for working memory. So what we're doing now is, you know, we're learning something new. Everyone's looking at their computer, looking at the images, processing information. This is the network that tends to fire when we're doing those tasks. And it never fires at the same time as the default mode network. Now, there's another network discovered by Seeley, which is the Salience Network. And that involves the anterior insula, the middle cingulate gyrus, as well as many other association areas, approximately 49 areas. And this network has a lot of importance in interoception, so understanding ourselves and understanding what it means to be someone else. But it's also got an important role in switching between the default mode network and the central executive network, because it's thought we're never in either one, we can never be in both networks at the same time. So this network functions as a switch between those two networks. The last network with 37 odd areas is the dorsal attention network, which involves the frontal eye fields on both sides of the brain and heavily in the interparietal sulci area. And that's important for focusing our attention. So the example I like to give is, you know, everyone's looking at their computer screen and not looking at something that's behind their computer. It's your dorsal attention network which is able to guide your attention towards that. Now, understanding these networks is the next step to understanding the brain. We've gone away from saying, you know, the lateral frontal lobe is just responsible for working memory, for instance. It is, but there's a lot of other parts that integrate with it to perform that function. All right, so with this background, and I know it's a lot of information to take in, based on this, it's an era of research explosion where people are studying diseases from brain tumors to mood disorders to traumatic brain injury to autism, all through the eye of the connectome. So what I wanted to do in the next part of the talk is to just look at some of the more recent developments in terms of concussion and mild traumatic brain injury. in relation to the connectome. So what studies have been done? There's not a lot of studies, and I'm sure in the next few years there's going to be an explosion of data that's going to come out. But starting from basics, I mean, what is concussion, which you all know better than me? So it's a form of mild traumatic brain injury with transient behavioral changes. And one of the defining characteristics is that there's an absence of structural abnormalities on clinical neuroimaging, but that just applies to standard clinical neuroimaging, because if we're starting to look a bit deeper in terms of the diffusion imaging, which looks at the axons, if we're looking deeper in terms of the functional imaging, such as the resting state functional MRI, there definitely are changes which we will explore. So if we broaden this definition of standard clinical neuroimaging, we can actually find abnormalities which can potentially explain why people are getting concussive symptoms or why they're persisting for some patients. Clinical presentation, there's a whole host of different presentations in concussion, from somatic complaints to problems with understanding, disturbance in mood, or disturbance in sleep, which is very common. And up to now, what we've relied is a standard MRI, which looks for any major structural problem. If you don't see anything, then you don't call it a traumatic brain injury, and you just tend to call it a concussion. And our understanding of it is based on clinical scales, such as the sports concussion assessment tool, or many other different scales. And they're used basically to try and prognosticate people, understand how bad the concussion is. And they're also used to try and get an understanding, is it safe for me to return to play sports? And as a neurosurgeon, I basically find it terrifying sometimes to have a patient with a normal brain scan, but it's explaining lots of symptoms, and they know they're not normal. You can see in their eyes they're not normal. And you've basically got an MRI scan, which is normal, and now you have to tell this patient, is this going to go away? Are they safe to go to work? Will they be normal again? These are all really difficult questions to answer. And this is where I think having detailed connectomic knowledge will be the future in terms of getting us more data for these patients. All right, so the first paper I looked at is Connectomic markers of symptom severity in sport-related concussion. So what they basically did, so this group from Canada, they had 70 university athletes, 35 of them were concussed, and then they had 35 age-matched controls. And what they did for all of them is they did detailed imaging, so resting state functional MRI imaging within 7 days of their concussion. All athletes had a SCAT3 baseline assessment, and they were also reassessed following their concussion, and they also had a battery of other tests. So just looking at the two different groups, we've got our control group and our concussion group. The age was identical in both groups because they were age matched, as was gender. And as is self-evidently, the concussion group had 1 concussion and the control group didn't. In terms of their concussion scores, so the control group had, very low symptoms, and this was the same for the baseline concussion group, i.e. the test they did before they got their concussion. However, after the concussion, it can clearly be seen that they had a much higher severity and a much higher number of symptoms. They looked at other aspects in their testing, such as orientation, concentration, etc., but there wasn't much difference between the scoring of the groups. So the first thing to do is to actually analyze the resting state functional MRI and look at the connections between the entire brain for patients with concussion or athletes with concussion versus athletes that weren't concussed. And what we're actually looking for is differences between the two comparisons. So when we're comparing the control group with the patients that had concussion, it became very clear that there was decreased connectivity in large parts of the temporal lobes and large parts of the frontal lobes. So if you're going to answer the question, if we look at resting state fMRI, can we tell a difference between someone who has concussion and someone who doesn't? The answer is yes. We can see it in their frontal lobes and in their temporal lobes. What was also interesting is that converse to decreased connectivity, the cerebellum actually showed increased connectivity, almost like a compensatory mechanism to the concussion in the athletes who had concussion versus the control group. Now, the next thing that this study group did was, in the previous slide, they just looked at the entire brain. So they looked at the resting state functional MRI signal. In the next part of the brain, they actually defined brain networks. They didn't describe the networks the way I described them earlier. They sort of took a more anatomical approach in terms of a frontal lobe circuit, a temporal lobe circuit, a visual circuit, etc. So they made predefined networks, and they wanted to see within the network how much connectivity is there. And what they wanted to do was associate this with concussion scores. So in the middle graph, what we have on the x-axis is a symptom severity, i.e. when we're grading the concussion, how bad it is. And then on the y-axis, we've got connectivity within the network, i.e. how connected is each part of the subnetworks of the brain that we've drawn. And what was fairly clear is, so H1, 2, and 3 here are patients with high symptom severity, and L1, 2, 3 here are patients with low symptom severity. And what was clear was that the higher that the symptom severity was, the reduced the amount of intra-network connectivity. So because we're low here on the y-axis, we don't have a lot of network connectivity, as opposed to lower concussion scores and normal patients. And in fact, if we look at the individual connectome analysis, so when you look at connectomics, you look at something called an adjacency matrix, and it just looks at the connections of different parts of the brain. But what we're actually showing here in different colors is how connected a network is. So if it's red, it's very connected. If it's blue, it's not very connected. So what we're seeing in these subnetworks, we've got lots of red for the patients with low concussion scores, whereas we've got a lot more blue for the patients with high concussion scores. And what that's showing is that at a local level, there's a lot more reduced connectivity of the patients with concussed brains. Okay, so the question of the first paper is, if we're looking at resting state functional MRI, can we pick up differences between someone that has concussion and who doesn't? And clearly, we showed that you can. So the next step, which is, it's a natural evolutionary pathway for anyone studying a disease in connectomics, is to use machine learning to try and differentiate between a patient with concussion and someone that doesn't. So the next paper from the US looked at detection of mild traumatic brain injury by machine learning classification. And what they did is they used both resting state functional MRI, but also that diffusion imaging. So they used two different modalities to try and see if one was better at predicting who had concussion and who didn't. And the way they did that is they studied 47 patients with mild traumatic brain injury. So this isn't technically concussion, patients that presented to casualty. They had a GCS of 13 to 15 on presentation. They didn't lose consciousness more than for 30 minutes. And they had a maximum of 24 hours post-traumatic amnesia. And they matched these traumatic brain injury patients with gender-aged matched controls. And what they did is they took the diffusion imaging from all the concussed patients, or from all the patients with mild TBI and all the normal patients, and they put it into a machine support vector learning protocol to try and see if the machine learning can pick up differences between patients that had brain injury and the ones that didn't. Then what they did is, for the second group, is they actually looked at the diffusion imaging, i.e. can the machine learning tell if there's differences in the pattern, the axonal pattern based on the diffusion imaging. And then they also mixed and matched both diffusion imaging and resting state functional MRI. In training the machine learning algorithm, what they told it to do was look at specific brain networks like the previous one. So they set some parameters and told it, I want you to look at the default mode network, the language network, the central executive network, the visual network, and then trained it to look at clusters of the brain that should be firing together. Now, what the machine learning told us is if we look at patients with mild traumatic brain injury and normal patients, we can build a machine learning algorithm that has about an 84-85% accuracy in telling the difference. So if you feed in a group of patients and the machine learning is blinded, and some of them have mild traumatic brain injury versus some that don't and are completely normal, you've got about an 85% chance of that machine learning algorithm telling the difference. And what they found was that the resting state functional MRI imaging, i.e. the bold imaging, was better than the diffusion imaging, i.e. that tractography where we're looking at brain tracts. So it was 85% accurate versus 75% accurate if we were looking at the diffusion imaging. They also tried a model which actually combined the two. So we're using both resting state and functional anisotropy, i.e. the diffusion imaging. And it actually wasn't better than, when you combine the two, it wasn't better than either one alone as one might predict. It was actually, again, around the 75% mark. The other two groups is just different methods that they use to deduce the axonal patterning based on the diffusion imaging. Now, interestingly, when we're looking at the areas that played the biggest role in terms of telling a difference between someone who had mild traumatic brain injury versus someone that didn't, interestingly, it was the left angular gyrus and the cerebellum, which came up again in this paper, like the previous paper where we said that there was increased activity in the cerebellum as a compensatory mechanism in the concussion patients. So where we are so far is the first question was, can we find differences between patients with brain injury or concussion and normal controls? And the answer is yes. Can we use machine learning to pick up the differences and build models to tell us who has concussion and who doesn't? Now, the last paper I'll go through is looking at actually subconcussive impact exposure in US football players, which is a huge topic. And what they wanted to do was look at the default mode network, which I described earlier, and said that it was responsible for internal thinking, internal mentation. There's quite a few papers that have come out to show that that's one of the networks that's predominantly involved in post-concussive encephalopathy and a lot of the long-term sequelae to head injury. So they wanted to study how this network functions in patients with subconcussive events. So the question that they were looking at, does American football related repetitive head impact exposure lead to aberrant DMN or default mode network connectivity? And the way they studied this was they had 50 young males between the ages of 8 to 14 who played American football in the US. And what they did is they did both pre-season imaging and post-season imaging for these adolescents. Also, what they did for the teenagers in this study, they fitted their helmets with impact telemetry systems to actually record acceleration and deacceleration events. And by doing that, they were able to deduce a number of head impacts during the season. And what they did is they compared this group of patients or athletes to 20 age-matched non-collision sport controls, i.e. 20 athletes who were playing basketball or tennis or something that didn't involve impact. And those were matched for age and gender as well. All right, so what did they find? So the first step was pretty easy. They just said, okay, let's study the default mode network in our control patients. And we know where it is, i.e. it's in the cingulate gyrus behind the corpus callosum, the interparietal sulci, the medial frontal lobes, and a little bit of the temporal lobes here. And what they said is, how does the default mode network compare in our tennis players and our basketball players compared to a post-season MRI from an adolescent that's had impact trauma during their season? And what we find here is that we've got a less pronounced default mode network. And in fact, this image to the right with the blue areas shows the areas in the football players that didn't come up as often as the non-contact sport athletes. On these graphs here, what we're doing is we're comparing the athletes in non-impact sports, such as tennis in the blue, and then we're comparing the football players in the red. And what we've got is we've got a pre-season MRI and then a post-season MRI. So in the pre-season MRI, where these children didn't have any contact, there wasn't any significant difference, which is shown by little stars, between the default mode networks of football players versus non-contact sport players. We've only got one area. So each one of these little columns is a different area on the brain. So this could be left interparietal sulcus, this could be right interparietal sulcus, this could be cingulate, gyrus, and so on and so on. What we've got in the postseason, so now these young American football players have had a whole season, and we've repeated their brain scan after the season of impact. And what we're actually finding is that there's a significant difference in multiple areas that define the default mode network between contact sport athletes versus non-contact sport athletes. So clearly the default mode network is getting strained. And this could have serious future implications for these adolescents. So what can we see as a conclusion? So I think there's a very promising role for Connectome and the metrics that we can get, and it becomes a lot more deeper than what I've presented in terms of making biomarkers for concussion. So not only will we have behavioral scores that we can do, but we can probably build models where we scan patients and are able to come with some sort of metric that tells someone how bad is your concussion and what is your future risk. It needs a lot more data, but it's certainly possible. There's a potential role for the default mode network, which we talked about in the last paper in terms of determining how bad concussion is. Without a doubt, machine learning is the next step, and it's going to provide data-driven and personalized medicine to this problem and many other problems. And ultimately, what I said earlier is, we are on the pathway through this technology of building a prognostication models based on the network analysis. Okay, so why am I interested? So I'm interested because basically I'm a brain tumor surgeon, and as John alluded earlier, I did do a fellowship overseas, and I was trained by a world expert in connectomics called Mike Shagru, who subsequently founded Omniscient, which is a brain mapping company. And I'm interested because very often I'm called to take out very difficult tumors in my patients, which are destructive to the brain. And I have to try and take the maximum amount of tumor without damaging patients. And this can be very difficult. And there's a huge role for data science to improve resections and patient outcomes. Now, what's very interesting is during my time with Mike Shagru and Omniscient, I also got introduced to a technology called transcranial magnetic stimulation. And what that's able to do is use different brain frequencies to stimulate parts of the brain or suppress parts of the brain. And what we're actually doing is we're understanding brain connectomes and brain maps in the context of different diseases. And we're able to see which parts of the brain are upregulated or downregulated. And we're trying to intervene for neurorehabilitation with transmagnetic stimulation. Now, what we've actually got in terms of trying to understand how the connectome maps work is what Omniscient's done is for any patient, you can get an MRI scan and basically use machine learning to break down their connectome, and then identify different areas based on that Glasser parcellation scheme. And then what you're able to actually do is look at their connectivity and compare it to a cohort of young adults with no neurological problems. And we're able to visualize different parcellations or parts of the brain, as we're showing here on an adjacency matrix, and figure out if it's red or if it's blue. And that lets us know if it's overconnected or underconnected. So with this connectome technology, we're actually getting a lot more data in terms of individual parts and how they're functioning. So for instance, this is a connectome from someone with a language problem. And in a normal person, you will just get white and black. whereas the red and blue areas show us different parcellations or part of the brain with abnormality. And that we can use it to make plans for intervention. I'll just go next here. So as I alluded before, what transcranial magnetic stimulation does is you've got a circular magnet with a figure-of-8 coil, and you're able to make a magnetic field, which becomes an eddy current, and that penetrates the surface of the brain. And based on the frequency or how you use it, you've got an ability to overstimulate, to hyperstimulate the brain or suppress it. And that brings long-term changes. So this is, it's just an example of why I'm interested. So this is a patient with a deep brain tumor that basically was in the sensory cortex, and they had problems walking after I resected it, which is expected. So what we see here is a white area in the MRI, which is showing a low-grade tumor. That's an axial scan, that's a coronal scan, and that's a sagittal scan. And on the right, a post-op. So the entire tumor is moved, which is good because this can change to a glioblastoma. And if that does, then the patient only has a 1-year life expectancy. But the problem was, by taking it out, we did have a clumsy foot, which the patient had pre-op, but it was worse after taking it out. So what we did was, okay, let's understand this connectome better. Which brain parcellations are abnormal in the sensory motor system, which we can filter out. And let's build a plan of TMS to try and improve that. So we did do that. We found some targets based on these personalized maps. So this was the patient's foot before TMS. And on the right, this was her foot 3 days after TMS. So you can see here, it's finding it very hard to find the pedal, can't orientate it, unusable. Whereas once we rehabbed it and improved the function of the motor parcellations, the foot became a lot more fluid. This is another patient with a really deep cavernoma, a type of vascular brain tumor, which had bled, was life-threatening and needed to come out. But being very deep in the brain and in the thalamus, which is critical, it was very difficult to get to. So we used the diffusion imaging and connectomic data to make an ideal approach to remove the tumor. But nevertheless, after this entire ordeal, she did have weakness on the left side of her body. So what we've got here is the axial and the coronal and the sagittal image of a cavernoma before the operation, and that's after the operation, so it all came out. But we did have the deficit to deal with. So again, we use this understanding of the connectome to try and make a plan to try and rehab this patient. So pre-TMS, she was basically not able to walk independently. And then, but by the first week, we got her independent. And then this was several weeks after, where through the neuro rehab and trying to stimulate the correct areas, we got her walking independently and fluidly. So this is why I'm excited about the field, but it has tremendous applications to any part of neuroscience or any neurological condition, any neurosurgical condition or psychiatric condition. And I think the future will give us a much deeper understanding in terms of what concussion does to the brain, and also potentially help us prognosticate and hopefully maybe even give us ways to intervene. Thank you for your time.
Chris, thanks for an absolutely fascinating presentation. It wasn't easy to deal with such a massive topic to which many of your participants on this webinar would not have been exposed. So you really handled that magnificently and gave us a great introduction into the science of connectomes, and then also the application to sport-related concussion, which, as you alluded to, is really in its infancy and just showing its potential. And I think that's what excites me so much about this. So as expected, there have been a number of questions which have popped up, and if you wouldn't mind just spending the next 15 or 20 minutes with us to answer some of those. So the first questions really relate to accessibility, and cost in South Africa in particular, both for the imaging technology and also for TMS. Can you just give us some idea of the availability, and maybe also internationally, you know, is this becoming something that's increasingly accessible, and is it affordable?
So one of the huge problems with this field was, outside of Harvard University and Ivy League setups and, you know, Cambridge in the UK and a few setups like that, it was really difficult to analyze the connectome if you didn't have a whole data science department behind you. And the problem with that was, like, you know, most normal people, clinicians, would have no idea how to look at the data. Where we've been lucky is my ex-boss, Mike Shagru, and Omniscient have made a platform which any hospital in the world can take a MRI and build the basic sequences for brain mapping, submit it to their platform, and extract raw data to analyze. So the cost at present is, if it's someone with a neurological problem and they have a medical aid, you can get the MRI on the medical aid at no cost to the patient. If they don't, currently at Linksfield, the price is about 5,500 Rand to do the Connectome MRI, and then analysis of the brain mapping is around 3,500 Rand. So look, there is a cost involved, but there's a lot of you know, steps involved in analyzing the connectome and getting the raw data. What we're also doing is we're training clinicians. So if clinicians are interested in understanding more about the connectome, we've actually got a training program, and, you know, we're excited when doctors want to learn more about the connectome and how they can start analyzing it and using it in for their clinical use. For instance, at the moment, we've got quite a few psychiatrists which have been trained and able to use it for patients with mood disorders, etc.
Great. And TMS, in terms of its accessibility, is it becoming more mainstream? Are the psychiatrists, for instance, using it on their patients a lot more?
Absolutely. So where I was in Sydney, there's a lot of different TMS clinics. There's very few clinics which are using navigated TMS, i.e. using sophisticated brain maps to try and find abnormal areas, and then using brain navigation to target the TMS to specific areas. Based on when I came back here, I really wanted to continue this field with all my rehab patients for neurosurgery with brain tumors, brain trauma, etc. So we have set up Connectomics with Netcare, and we do have a TMS set up in Johannesburg, and very soon there'll be another one in Cape Town. So we're trying to make it more accessible for patients, but also doctors that want to be trained in the platform and bring their own patients and make their own brain maps and TMS plans.
Increasingly in concussion, we're viewing them in terms of different subtypes or related to the different domains that are affected. So, for instance, you may have a more cognitive type of presentation, a more vestibular or ocular presentation, more emotional, even autonomic. Now, it looks to me, with this more elaborate network and map of the brain, that this is custom made for looking at that sort of subtype of concussion. Do you see this as giving us a lot more insight into those sort of areas that are involved?
Absolutely. So what we're able to do, so the one thing that we're doing is, the way we can analyze the brain is, any patient that has concussion, we can break it down into a Glasser Atlas, which is 360 brain areas, which is unique to every human being because everyone's got different gyri, different sulci. But because the machine learning can break down the exact area, we can compare it to normal or control patients. Now, what's interesting is we've also mapped, you know, where is the default mode network, where is the Salience network. So if you're telling me a more cognitive aspect, then I want to see changes in my central executive network. If I've got autonomic disturbance, my Salience network is very important in communicating with the autonomic system. So I'm going to look for changes in the autonomic networks. if I've got predominantly visual problems, the visual network is mapped. We can look at that and analyze it. So the tools are there to absolutely break down the subtypes of concussion and look at them into a lot more detail.
And then we've got a few questions here from one of our leading sports physicians, Dr. Lee Pillay, and he's actually asked a non-sporting question that's related to ADHD and evaluation of patients with attention deficit disorders. Is there scope for assessment of those patients?
There is. So look, we're looking at a whole host of things, and like we did a talk for concussion, we can do a whole talk on ADHD and understanding the different networks and how they're affected. So the short answer is absolutely. There's people that have done TMS on an experimental basis with mixed results. Some have found improvement, some haven't. But I think we're in the very beginnings of understanding how the connectome shapes all neurological disorders. And the next decade is going to be an explosion in the field in terms of the data we get from looking at large cohorts of patients with a different conditions and understanding how the connectome is shaping that condition.
And perhaps in a sporting environment, if I summarize some of the questions coming through here, the step before that might be to build a library of baseline data. So for instance, we don't really know what the brain looks like in someone who's taking, as you alluded to, subconcussive impacts in contact and collision sport, but not presenting clinically with any symptoms. And perhaps we need to scan large numbers of brains of people who are not involved in collision sports, those who are, but who are asymptomatic and have never been diagnosed clinically with a concussion, and then compare those obviously later to those that are clinically diagnosed. So it seems to be that there's huge scope for research in this field, and then probably take it on into longitudinal studies where we follow these athletes down the line, because you are, I'm sure, aware that a big field of interest now is potential dementia in athletes who've been exposed to multiple collisions. And this may well be something that helps us answer those questions. I'm not really posing a question, but I'm asking you for your opinion, whether you agree.
I completely agree. I mean, it's all data-driven science. The more data we have, the better models we have, and the more scans we have over years, over decades, the much more complete our understanding will be with PTE and things like that. So completely agree. You understand the potential 100%.
We've got a question from a young neuroscientist here, and she's asking, as someone who's interested in this field, she's a machine learning engineer, very fascinated by neuroscience. As a South African, what's the best way she can contribute to this field and actually get into the connectome science? I mean, I can certainly put her in touch with you, but just give perhaps some idea of the scope of a neuroscientist becoming involved in this field.
I think it's basically, unfortunately, in South Africa, we don't have the infrastructure. So all the data science is done by Omniscient in Sydney. I'd be happy to communicate with them, and please communicate with me and see if there's any interesting projects that we can consider in South Africa in conjunction with Omniscient. Unfortunately, look, the field doesn't exist in South Africa and neurosciences, and unfortunately, a lot of the basic research we're looking at is fairly outdated, and that's got to do with resources. But please get in touch with me, and let's see what we can do.
Is there any data, or are there any data regarding sex differences on the connectome? Are there differences male to female?
No, not yet. No, we haven't found the pacillation that makes you male or female. Look, there's a lot of different ways. So currently, we're looking at the brain through the Glasser Atlas, but there's a lot of ways to even subdivide the Glasser Atlas into further fractions. You can actually look at time sequence. So right now, we talked about a static connectome, but there's a whole concept of a dynamic connectome, like how much time do you spend in a DMN versus CEN versus Salience. And then you can come up with brain states based on these networks. You know, some people say 5 brain states, other people say 7 brain states. So the data is infinite. I'm sure we can narrow it down with more data, but at present, I'm not aware of any study.
Okay. Do you see the potential for, as we do in, for instance, computerized neurocognitive testing, and even the SCAT tool testing, doing pre and post season or pre and post event testing, so analyzing or assessing a baseline for players? I presume that might be the basis for research, but also have clinical applications.
So, you know, if we're going to look at the Connectome, we need clinical data and we need ways to assess people, because that's what you're going to use to try and decipher differences. So absolutely. Omniscient is working on a very detailed neuropsychological battery of tests, which they're linking to the brain scans as well. So that could be an interesting tool that can be integrated. But yeah, absolutely. Like, we do need testing on a yearly basis, probably pre-season, middle season, and post-season.
We've got a few cynical comments about potential scanning of the referee in the Springbok game last Saturday, but I won't elaborate on those from some of our more cynical colleagues. But your presentation was that good that I think you covered many of the other questions that have come through here. With your permission, we will post the presentation on our website, the YouTube channel, so it becomes accessible. I think it's the type of presentation, if you view it 3 or 4 times, you actually start getting a good idea of what's going on. We will put some of those clinicians and neuroscientists in touch with you, and hopefully we can build the Connectome message and the Connectome network in this country, and try and feed into the science and into the research. And above all, I suppose, what we all aim to do, and that is to help patients, and in our case, help athletes with traumatic brain injury. And I think this has given us a great insight. It's opened a new window for many of us, and we look forward to continuing that communication with you, and looking to you to hopefully lead us in this area in South Africa. So Chris, thanks very much for the time. We really appreciate it. Thank you to all of you who signed on. We really had an outstanding attendance, as I said, from around the world this evening, and I'm sure you'll all agree it's worthwhile. Not one of you dropped off the presentation, so that's always a good sign. Thanks very much to Robin Saggers for being there in the background again. Thanks to our sponsors, Asina Lita, and all the people who support the WiSH network. Robin will put up some slides about upcoming presentations. And also, you know, once you signed on here, you will see how you can get your CPD points there. There's a link there. If you just give us a week or 2, you will receive your CPD certificates for attending this outstanding presentation. So thank you to all of you. We look forward to welcoming you at our next presentation, which is this spine evening, co-hosted with the Johannesburg Orthopedic Sports Institute and the Center for Sports Medicine and Orthopedics, another fascinating seminar. And I also know that the Tendon Interest Group is hosting an interesting discussion tomorrow on the rotator cuff. So look out for information on those on the social media posts. Thank you for attending this evening, and we look forward to hosting you in the coming weeks again. Good evening.